So, ladies and gentlemen, welcome to Doordarshan's special conversation with Dr. Stylianos Antonarakis, and he is a scientist, leading genetic scientist from Geneva, and has been working on Down syndrome for many years. So, Dr. Stylianos, I would like to ask that, if in layman terms, if I would ask you that, what do you do, and what kind of work that you do in Geneva in the genetic field, and how does your entire career progress is there, and what kind of things that you encounter, the truths, the ugly sides and good sides and everything. So if you can tell us your story in a nutshell.
Well, first, I thank you very much for the invitation. It is a great pleasure for me to be in Ahmedabad. I started a long time ago, actually. I'm an old-timer in genetics. I'm a medic who loved research too. So when I was finishing medical school, I said, what should I do now? And things that were interesting to me were the things that were unknown. Because I thought that the medical practice that we practice every day, it is practiced on things that are known. But if there's no research and there's no progress, medicine does not progress too. So I said, I'll go to research. And then I had to see which was the main question at the time. And there were two main unknowns at the time in the late 70s, to orient you in time. And the two unknowns were the human genome. What is the genome? What is a gene? What the genes do? How disorders are caused by genes. That was a major question. And the other question was, what is the brain? Can we understand the brain? So I chose to go to the genes and genetics because I thought that the brain was very difficult and it was not the right time to study it at the time. So I had a career in two institutions, the first in the United States at the Johns Hopkins University in Baltimore. And that was a part of my career for about 15 years. And then the second part of my career was in Geneva, the University of Geneva. But in both places, I studied the genome and the variation of the genome and how that variation causes the different traits and the different disorders that we all have. So that, in a nutshell, that's my career until now.
Correct. But you loved the puzzle of chromosome 21. So how did it interest you?
Yes.
And why did you choose that to solve that puzzle?
Right. So one of the most common conditions in most common genetic conditions in humans, it is called trisomy 21 or Down syndrome. And let me step back for a minute to tell you about the genome, and then I go to trisomy. So the genome is let's compare it to a very big book of instructions, a book that tells you how the cell works, how the human develops, how we grow, how we age, and which disorders we get. So that tells us a lot about who we are and how we interact with the environment. So there was a big project that started 1st of October of 1990 to sequence the genome. Sequence means to read the genome. The whole genome was unknown. We knew that existed, but we did not know the whole anatomy of it. And so the big project was conceived, so I worked on that project. And the genome is, as I said, is a book written by 4 letters, A, C, G, and T. And how big is the genome? 3 billion letters. And we inherit 3 billion from our father, letters and 3 billion from our mother. So the whole thing that we have in each and every one of our cells is, there's a book that has 6 billion letters. And those 6 billion letters, we pass them from one cell to another and from one generation to another. And the 6 billion letters, they are packed in let's say, 23 buses, big buses and small buses that transfer the genome from one cell to another and from generation to generation. The buses are not the same size. There are some big ones and some small ones. And we give them numbers. The very big one, we call it bus 1, chromosome 1. And bus number 2, 3, 4, blah, blah. And the smallest one, it is the chromosome 21. Chromosome 21, that has about 45 million letters, text. And you and I, we have 2 copies of chromosome 21, as we have 2 copies of every chromosome. But our co-citizens, the people that have trisomy 21 or Down syndrome, they have 3 copies of chromosome 21. Usually, 2 inherited from the mother and 1 inherited from the father. So in order to understand Down syndrome, we had to read the chromosome 21 as we had to read all the other chromosomes. So that brought me, the reading of chromosome 21 brought me in the trisomy. And the reading was finished in the year 2000. So year 2000, we knew the majority of the text of chromosome 21. Not all of it, but the majority of it, the most important of it. And then the next question was, can we understand that text? And of course, we do not understand all this text. We understand only the part of the genome that contains genes, because the genes that make proteins are the part of the genome that is probably quite important for our health. Not that all the other region is not important, but we focus on the genes first. So we had to know how many genes are on this chromosome. Let's say 150. We had to identify all of them, find them, see what they do. And then the next big question, which is still unanswered, is which ones of them, of these 150 genes, are important for the signs and symptoms of Down syndrome. The short stature, the intellectual dysfunction, the characteristic facial features, the heart defect that some people develop, and so on. So that took us a long time, not only people in my laboratory, but in the world's laboratory. And there was a collaboration between physicians, doctors that see patients, researchers, of course, the individuals with Down syndrome and their families, and also the funding agencies, the agencies around the world that support the research. So now we have ways to identify trisomy 21 before birth. We have ways to identify all the signs and symptoms of trisomy 21. We have identified 1 or 2 genes that are important for some of the features of trisomy 21. And now most people work on understanding exactly the mechanisms of the intellectual dysfunction and other features of Down syndrome. Because the big question is, can we treat? We can do prenatal diagnosis and tell the family that your fetus, your embryo has trisomy 21, or your newborn child has trisomy 21, but we cannot treat. But there is hope, because now that we begin to understand what is wrong with the mechanism of the trisomy 21, we can intervene. Because the treatment works very well if we understand the cause. That's why there is a lot of effort in academia, in the universities and the research institutions to understand the mechanism. And then the next step is to find treatments. But the treatments are usually the work of the pharmaceutical companies or the industry. So the collaboration between the academia to find the cause and the mechanism, and the industry to find treatments, it should be very strong and very collaborative in order to go from the cause to treatment.
Okay, correct. But in this study, did you find any particular gene type which are susceptible for this kind of mutation which will lead to the trisomy 21? So, can we predict it by reading at someone's DNA right now?
As I said, we can diagnose 100% of if a fetus or a child has trisomy 21. We can diagnose the syndrome.
But before that, can we, from a person's DNA sequencing or something, can we look up at their genetic makeup, and can we predict that this person's child might get trisomy 21?
This is a great question. This we cannot do. We cannot predict that a person's DNA, in collaboration with the other person's DNA that make a couple, could produce a child with a Down syndrome. That we cannot do. There's no way to predict if an individual will have a child with a Down syndrome. The only predictor at this stage is the age of the woman, because we know that as the woman becomes older and older, the probability of having a child with Down syndrome increases. So a young woman, let's say between 20 and 30, has a baseline risk, and a lady from 30 to 40 has a much higher risk, and a lady of more than 40 years of age has a much higher risk. But this is a population risk, not an individual risk. A couple cannot come and say, can you tell me what's my probability of having a child with Down syndrome? We cannot do that now. Perhaps in the future, perhaps never. Because there are two opposing hypotheses. One hypothesis is that, as you're suggesting, there's a predisposition. Yeah. And the other hypothesis is totally by chance. It so happens that when chromosomes 21 are separated to give a child, then something happens to that mechanism by chance, and the two chromosomes stay together, they don't separate, and then you have a child with a Down syndrome. The other hypothesis is that the chromosomes that do not separate, they have something on them or elsewhere in the genome. That we do not know. It is completely unknown. People tried to find an answer to this. My laboratory tried 30 years ago, actually. And we did not find anything, and nobody else has found anything like this.
Correct. So, can we do any intervention at the fetus level or development level if we find this kind of chromosome? Is there any gene editing is possible, or gene therapy is possible? If there is any way to come out of it?
Well, unfortunately, the treatment of Down syndrome is behind the treatment of other disorders, mainly because the Down syndrome is not a disorder of one gene. It is a disorder of one chromosome, which is a big chunk of DNA. It has, as I said before, chromosome 21 has 150 plus genes, and many other genomic elements that are very important for life. The whole chromosome is there, And it's quite difficult to have a treatment for a gene that contributes to the signs and symptoms of Down syndrome, which is now unknown. However, there's a lot of research to identify genes that contribute to the syndrome. For example, there's a gene that has a Kabbalistic name, DIRK1A, in our lingo, that we think, after the research of the last 10 years, we think that 3 copies of that gene is important for signs and symptoms of Down syndrome. And there are efforts in companies and in the academia to block a little bit the action of this particular gene, to lower down a little bit the level of the protein that is encoded by that particular gene called DIRK1A, and therefore introduce a treatment to trisomy 21. That's very hopeful, and I hope that this kind of treatment will mature. It works a little bit in mouse models, because a lot of research that we do in humans, we cannot do it in humans actually. We do them in animal models. And the best animal model for trisomy 21 is a little mouse that has 3 copies of the equivalent of human chromosome 21. And there are several of those, several generations of those, and the scientific community are using them. So the best friend of individuals with Down syndrome are not the doctors, but the mouse.
Correct.
The mouse that has trisomy 21, so we can experiment therapies and ideas of how to treat that mouse, and then transport that treatment to the human.
So how is your friendship with the mouse?
How?
How is your friendship with the mouse throughout these years?
Mouse is a good friend and a good teacher, and we learn many things. And we learn the function of genes, we learn the interactions of genes, we learn some proteins that are important. And the interesting thing about the mouse is not only to understand the mechanism, but also to have experimental therapies. And one has to have many experimental therapies, let's say 100, and perhaps 3 of them work. It's a trial and error based on the scientific knowledge and the prior probabilities. But after this, it's a trial and error. You don't know if something will work or not work. So that's why I said that the mouse is the best friend of trisomy 21 individuals.
Yeah. You grew up in a post-World War scenario in Europe, and also seen the beginning of 21st century. And we are now near the ending of the first quarter of 21st century. The genetics in your times might be different. It has grown in a very different way. So, as a very senior person in this field, how do you see the entire journey from 70s to this many years? You might have seen the PCR also getting Nobel Prize, and you also see the CRISPR technology getting prizes also, and you also see same year you are seeing the stem cell therapies and gene therapies for sickle cell anemias in UK and USA. So how do you look back to the entire progression of genetics in these years?
I would say with one word, I was very lucky. I was very lucky to work on the genome and genetics in that period of time between, let's say, the end of the 70s up to now. Because my generation of scientists and physicians were proud to sequence the human DNA for the first time, the Genome Project. We're proud to identify almost all the genes. We're proud to identify most of the regulatory elements of the genome. And we're proud to crack down most of the so-called monogenic disorders due to a mutation or a variant, as we say now in our vocabulary, variants in single genes. And we talked about Down syndrome, but our laboratory has worked on several disorders due to single genes, starting from thalassemia, sickle cell disease, hemophilia, cystic fibrosis, and many other disorders, including many conditions of intellectual disability. So I was very lucky, and let me tell you, it was a roller coaster of excitement. Every year there was an excitement of new things. So you hear very often today that, we have a bright future. I'm telling you, in 1970, we said we have a bright future. At the 80s, we said we have a bright future. In 90s, the same. In the 2000s, the same. 2010, the same. Every year and every decade, it was such an excitement, such an important joy to be in that field. And I do not see the end of it. I think it will continue to be like this in the next 50 years, because we have to answer, or the next generation has to answer many questions. What causes disorders? What's the mechanism of disorders? How the gene is involved in the human conditions? How the genome and the proteome interacts with the environment? Why some people with a bad mutation develop a disease and somebody else with the same mutation does not develop a disease? In our language, we call this penetrance. That's a mega question. What is the mechanism of a penetrance? And then, once we learn all this, how to treat. Because the people out there, our brothers and sisters and our sons and daughters and our mothers and fathers and ourselves, we care about treatments. But treatments do not come down from the sky. We have to understand and then treat.
So where do we see the zenith of this journey, or the last station of this journey? If still we are talking about the bright future, what would be the brightest point in the future where we will say, no, we have reached to the ultimate understanding of genomics?
That's a difficult question, because the human disorders that are due to the genome, there are thousands. And for each one, there's a turning point moment. For a very rare disorder that affects 1 in 100,000 individuals, the turning point is when we identify a treatment for that particular disorder very early on, and to treat the cause and not the symptoms. So for each one of them, there's a turning point. I give you an example. Let's take a disorder of blood coagulation that's called hemophilia. The turning point in hemophilia was when the gene was identified and recombinant protein factor VIII was produced because of the genetic engineering. And when this protein is given to individuals, and we treat them. We do not treat them by gene therapy. We treat them by an exogenous protein that is identical to the human protein. So for each disorder is different. It's like you ask me, what is the most important point for humanity? I will ask the question differently. What's the most important point for you?
The zenith point of...
And for you, perhaps, is when you have your first child born, or when you got married, or when you got your degree. So, for people, the important point is the treatment point for their particular disorder.
Correct. So I would like to ask that the way we understand the genetics, so has it changed the perceptions to see and look at the human body in these many years? And you also deal with genetics and intelligence. So have you really find out what exactly is intelligence? like what we really call, don't you think that some person who has a genetic mutation might have a different kind of intelligence than what you and I may have, but we are not able to see it because their way of functioning or interaction may be different from ours, but biologically they are surviving. So what's your opinion on this?
So intelligence, as every other biological determinant, has a variation. And most people are between point A and point B in that variation that we call normal. So there are normal people with high intelligence and normal people with less than high intelligence. But they are all within the normal range. There are some individuals that are way outside the normal range. They have a very much lower intelligence than the mean. And the reason why that is because there are variants, mutations, in some genes. I said some, but I would say many genes. There are probably 2,000 genes that if they have mutations, each one of them, they result in intellectual dysfunction. So the level of cognition definitely depends on the different genes in our genome, definitely. Of course, the level of cognitive function also depends on the environment. I give you an example that every student of genetics knows. There's a rare human disorder that's called phenylketonuria, in which people in the blood, they have a high amount of a substance called phenylalanine. Why that is? Because they have a defective gene that produces a defective protein that does not metabolize that chemical. And that's a genetic disorder. Now, if you have that genetic disorder that's called phenylketonuria, and you drink milk without that substance, phenylalanine, or food without that substance, phenylalanine, you develop normally. If you eat food with normal amount of phenylalanine, you develop an intellectual disability and neurological defects and other defects. All right. So that disorder is, you can look at it from both sides. It is a genetic disorder because the genes are defective, and it is an environmental disorder because you see it when you give the individual phenylalanine. So the intellectual level is the product, it's likely to be the product of the interaction between the genetics and the environment.
Correct. Another question I would like to ask, that you spent your entire career in Johns Hopkins and in Geneva.
Yes.
So you must have been working with the big names of medicine field and top-notch scientists also. So with whom you were associated all through years, who has inspired you, or whom you have inspired, or you worked together? So if you can name few names.
Few names,
yeah.
Yes. I was a student of Professor Victor McCusick. He was my mentor. And Victor was one of the founders of human genetics, not only in the United States, but in the world too. So I was very lucky that I was his student, and he was my mentor, and I learned a lot from him. And also the environment that he had created at the Johns Hopkins University at the time. Yeah. There are many others, but I will mention only him. And of course, the success of research laboratories depends on the caliber and the abilities and the ambitions and the ideas of people that work with you. And I think the secret is to to recruit people that are better than you. Because if you select people that are as good as you are or a bit less, you may have good control on them, but they will not contribute that much. Well, if you have better people, you probably have less control, but it doesn't matter in science. But they bring ideas, and they bring enthusiasm, and they bring alternative ways of attacking a problem. So if there's a message for the younger scientists or scientists is choose your collaborators well, and choose the ones that are better than you.
Can you elaborate this thing with some example or something from your experience without naming? So I think it would be very much useful for people to how to run an organization.
Yeah. Well, that is so common that I do not have any specific example in my mind. You know, when you run a sizable laboratory and a sizable clinic, and you have responsibilities, administrative responsibilities, either in the academia or a research center or a university or in the industry, the time that you need to think is quite limited, and is also contaminated by all these other things. So the pollution of all the other things that you have in mind and you have in your agenda to do per day. And then you go to the laboratory and you talk to your colleagues, and you hear a new idea, a new way of doing things, an alternative method, something that it's a bit unusual, a bit crazy, undoable, that you may say in the beginning, oh, that's nothing. And then you walk back to your office and you say, this girl may be right, or this fellow is probably thinking more ahead than me.
Then had you approached that person back and you approached that person and talked about it, or do you really utilize your authority and power to enable him what or he or she wants to do?
Well, of course, the first, because authority and power in science, I tell you, does not go far. Okay. It may go far in other fields, but not really in science, because the science is made by the agora of people, the collection of minds, and if you put the hierarchy on this, you kill innovation. That's my impression.
Correct. Doctor, how did you face the failures in your life?
As you know, when you do an experiment, 9 out of 10 times the experiment fails. So failure is the everyday life of a scientist. So it's not unusual. And you need to try again, and you need to understand sometimes, or most of the times, that what you did was wrong. And you need to change your strategy, you need to change your method, you need to change the way of thinking. And sometimes you need to abandon a project. There was a very wise mind in the UK, Sir Peter Medawar, who said that a scientist needs to study something that can be answered. Because sometimes you attack a problem that cannot be answered with the present technology or the present time in history. And you have to realize what is doable and what is not. And also you have to take calculated risks. A risk of 10% is acceptable. A risk of 100% perhaps is not acceptable.
So in your journey, any point of time, did you feel that the people around you are not understanding you, or you feel that what kind of work I am doing, which might not be recognized by the people who are surrounded, non-scientific people, those people who are most interested in FIFA matches, more interested in politics, more interested in Hollywood films. So didn't you feel that the things that you're doing for greater good of mankind and the mankind itself is not able to appreciate and understand what you're exactly doing. Have you ever felt that any point of time?
Well, look, when the people that count are your peers, and you have to convince them, and then convince, of course, the funding agencies, but the funding agencies are your peers too. So you need to convince your peers that what you want to do is important, innovative, useful, and the opinion of others do not really mind that much. Do not really count that much, excuse me. I did not tell you, but I was born in Greece and raised in Greece, and I was bombarded with the classic education of ancient Greece. So there was one philosopher, Socrates, who said, "Do not worry what the majority of people say, but you need to worry of what the people that know say." So that's what I'm saying. It's useless to pay attention to what the whole society in general says about something, because you don't share the same knowledge, you don't share the same goals, the same objectives. You don't share the same knowledge about technology, about what is possible, what is not possible, what is coming in the next 3, 4, 5 years. And the people that know this are your peers, the people that share the same experiences with you, and then you need to convince them. Now, sometimes you have an idea that you cannot convince anyone that is new and innovative. There may be a problem, because you need to, in order to do your research, you need funding. And as I said, funding comes from peers, comes from society, but it funnels down to whoever will judge whatever you have to do.
But do you really think that the more we know, the more we solve, or the more we know, the more problem we get into? So what's your experience regarding that?
Well, I would say the more we know, the more the more you get away from the other. Knowledge, I think, of course, knowledge unifies us, but knowledge separates us too. And we diverge. And then the effort is to put people together back, the so-called interdisciplinary type of research. You take a mathematician and you put with a biologist, with a physicist, with an ethicist, and all of them go towards a specific goal. But at the end, the idea, and that's my personal opinion, of course, the idea and the final objective, if you go from the generality to the specific, it's debated by the people that can debate this with appropriate knowledge and appropriate experience.
But do you think that all this genetic knowledge that we are gathering, we are experiencing, we are going through, will make us superior from other species without going through a natural process? And it will be detrimental to the humankind?
Well, I would not say we make superior from other species. I would say makes us more wise within our species.
Okay. Okay. Last question I would like to ask, that lots of young people in India are watching you. So what advice would you give to the young Indian scientist who is watching this interview from a remote village in Kolkata, in West Bengal, or remote village in Jammu Kashmir or Gujarat? So it will be telecasted to all over India. So I just would like to ask that what would you tell that person, that scientist might not be in the lab, that the scientist might be inside, the curious person who's seeking answers to the questions that is not letting him sleep every night. So what would you tell him?
I would say follow your dreams. I would say being obsessed with what you love. And in life, if you can, do whatever you like to do, whatever drives you. And have a good training. Try to get the best teachers, and if you go to science, you try to get the best pedigree, the best mentors, because it is very important to have excellent mentors, and then after that you develop your own contributions.
Thank you, Dr. Stylianos, for talking to us. It was really word of wisdom and a journey. And it was like a time travel from your days in Geneva, from your early days to these years that we have seen, and almost 50 years of your journey that we experienced over here. And I'm sure that our audience, our people, our viewer will be really inspired from that. Thank you so much for joining. Thank you so much.
Well, thank you very much for your invitation and your interest in science.
Yeah, thank you. Thank you so much.